Hello everyone. So this is another video from Dr. Surya Narayanan. So in this video, we will be commencing our discussion on the NEET 2021 biology question. So sort of analysis that we are going to do in this video. So to make it simple, in this discussion, we will be taking a chapter, a unit called as Biotechnology, which is from CBSE, NCERT class 12 concept. So this unit 9 biotechnology has got two chapters. There is chapter 11, chapter 12. Chapter 11 is basically about biotechnology principles and process, and chapter 12 is about biotechnology application. So in short, the questions that have come from these two chapters will be discussed in this video. Okay, so the questions, the solution, and its relevance to the NCERT text. Everything will be discussed in detail in this video. So I would, people who are watching for the first time, so I would like to request them to subscribe the channel for getting more updates. Do share with your friends and feel free to give your feedback. Okay. So first question. So and before that, I have taken this questions will comprise of both botany as well as zoology, and the order of the questions might vary from the one given in the question paper and the one which is discussed in this video, right? So with this, so let us first start with question: which of the following is the correct sequence of steps in PCR? So basically PCR is the polymerase chain reaction. So in the present pandemic, so the one word that we are all familiar with is PCR. How the test that we do to diagnose whether a person is COVID positive or negative is based on RT-PCR. So you have to include the word RT. RT stands for reverse transcriptase. Okay, reverse transcriptase. So reverse transcriptase is basically an enzyme which does the role of reverse transcription, right? So why is the reverse transcription required? Means the PCR generally accepts DNA, whereas the COVID is an RNA virus. So there is a need to convert RNA back to DNA. The need to convert RNA back to DNA. Hence the word reverse transcription. So this is all about a basic idea, okay, about RT-PCR. So coming to the PCR, PCR is actually a sort of amplification reaction, okay, with three different steps. Okay, so I think you have got the picture here. You have got the picture here. So the picture says that there is a denaturation step, there is an annealing step, and there is an extension step. So this comprises of one PCR cycle. Okay, this comprises of one PCR cycle, and this can be repeated. This can be repeated n number of times. So n might be 35, 40, depending upon our requirement. Okay, so this denaturation, annealing, extension, one cycle can be repeated n number of times, 35 to 40 times. So we tend to amplify a particular gene. So amplification means increasing its number, okay, from basically 2 from 1 DNA to millions of copies. So this is that's the reason why do we call it as a chain reaction. So this is a reaction, I hope you all know the meaning of reaction. So this is a chain reaction, means continuous reaction keeps happening, and this is done by the enzyme polymerase. Hence it is called as a polymerase chain reaction. So as the picture says, it's a denaturation, annealing and extension. So you have to go with the option denaturation, annealing, extension. So option B would be the correct choice here. Okay. So this is first question regarding PCR, and the reference here is this comes under from chapter 11, which is biotechnology principle. That is NCERT 12th book, page number 202, gives you this diagram, okay, which gives a clear message that denaturation, annealing, and extension, and which can be repeated n number of times to amplify our gene of interest. This is all the basics, right? So this is regarding the first question. Fine. So second question is, which of the following is not an application of PCR? So this is another question from PCR. So, but this application is an aspect of PCR is not from the biotechnology principle, but biotechnology application, for which we have to know what exactly the PCR is, which is what is described in chapter 11. That is biotechnology principle, right? So if anyone wants to know exactly what PCR is, there is a video already given in our channel, so you can very well go through that to have a clear-cut idea what PCR is. Okay. So coming to the application, so which of the following is not an application? So there are four options given. So they are asking which of the one which is not an application of PCR. So detection of gene mutation, molecular diagnosis, gene amplification, and purification of isolated protein. So already we have seen that in the previous question that the gene amplification is a principle of PCR. Okay, you try to amplify a gene to millions of copies, right? So it's basically amplification. So which means that this would be gene amplification would be one of the possible application, right? So this is what is used in recombinant DNA technology and producing insulin, hormone, everything. It's basically we try to increase the number of genes, and you do have the relevance here, right? So the amplified fragment. Okay, so again, this reference I have taken from chapter 11, and you do have a reference in chapter 12. So basically, this belongs to, ideally this fits to the chapter 12. That is biotechnology application. You have a line here saying that so amplification. So you can see here can be detected by amplification on their nucleic acid by PCR. So it is basically an amplification reaction. So mind, this paragraph I had given from chapter 12, and the previous one was from chapter 11. So mostly this question fit to the chapter 12 application aspect, right? So which means that this is going to be an application of PCR, right? So next is molecular diagnosis. So the very first question that I had begun, okay, would give you that molecular diagnosis will be an application of PCR. So what was that discussion? I said in present day situation, the pandemic, the one test that we have, the powerful test what we have, the most accessible test what we have, okay, for detection of COVID is RT-PCR. So which means that PCR can be used, okay, for diagnostic purpose. So before this COVID, for detecting tuberculosis, various other infectious diseases, so what all you need is to know that particular DNA of the bacteria, so with which we'll be able to synthesize the primer, and primer will be able to bind to the DNA. Okay, so we'll be able to confirm whether the person has got that particular infection or not, right? So molecular diagnosis is generally an application of PCR, and here also it is given. So the very topic is molecular diagnosis. Okay, so which means that I cannot go for the option B, which is an application of molecular diagnosis, an application of PCR, right? And detection of gene mutation. So gene mutation can be detected. So I hope you all know what mutations are: a change in nucleotide sequence in a DNA. So it is, see here you have it's a line saying that it is being used to detect the mutations in gene in suspected cancer patients too. So you could see that this question, even the previous question were all the direct questions from NCERT text, right? So we just need to understand these concepts and try to apply, so that attending these questions will be very, very easy. So gene mutation is an application, molecular diagnosis is an application, and gene amplification is also an application. Whereas the purification of isolated protein, PCR, doesn't have any role. So what is involved then? It will be generally some purification technique, like you can go for chromatography. Okay, you can go for chromatographic technique. So chromatographic is one technique that we have, general technique, preferred technique for purification of protein, but we do have some other technique. Okay, but generally this chromatography is preferred. So this is all regarding the question 2, and you can see the reference, okay, in page number 2 and 2 chapter is going to be the 12th chapter, that is biotechnology application. So these two questions that we have seen are a direct question from the NCERT text, right? Coming to the third question. Plasmid PBR322 has PST1 restriction site within the gene of AMPR. So AMPR stands for ampicillin resistance. If this enzyme is used for inserting the gene beta galactosidase production, and the recombinant plasmid inserted in E. coli, Escherichia coli strain, and they have given 4 possible options, right? So this is not a direct question. This will be some sort of an application question, and the relevant concept is given in our biotechnology principal chapter, that is chapter 11. So what is the relevant background information, relevant concept? So this comes under something called a selection process. Okay, so first I'll try to discuss the concept, then we will see the content given in the NCERT text. So you have something called as selection process, right? So in selection process, you have one way to select the recombinant cell, transformed recombinant cell, is insertional inactivation. Okay, insertional inactivation. So it's actually selection process is a very big topic, of which insertion inactivation means if already a gene is there, say assume that there is a gene already present. If I'm going to insert another gene, say assume that this is going to be the gene A, I'm going to insert gene B. Due to the insertion of gene B, gene A becomes inactivated, right? Gene A becomes inactivated. This is what is called as insertional inactivation. So I'll make it very simple. I'm not getting into the details. I said it's a very big topic to be discussed. So in simple words, there is a gene which is active. What do we mean by the term gene active? Means that the gene will be able to express its information into the protein. Now there is another gene which has got inserted to the gene A, due to which the gene A is inactivated due to the presence of gene B. So due to the insertion, and gene is inactivated, so which is what is called as insertional inactivation. So this is what the meaning in simple words, right? I hope you would have understood. Now, to understand this question, it is essential that if you have an idea of what PBR322, the diagram of which is given in our NCERT text, fine. Even if you don't know, you'll be able to manage it, probably if you have a basic idea, okay, with these concepts. So what do they say? PBR322 plasmid has got a gene which confers an ampicillin resistance. So this is going to be the ampicillin resistance which cleaves the ampicillin. So the one cell, okay, which will be sensitive to ampicillin will be resistant due to this plasmid, okay, containing this particular gene. So this is going to be ampicillin resistance, right? Or ampicillin resistance. Now, what is happening is this ampicillin resistance gene is being inserted with another gene, which is beta galactosidase, which is going to, again, beta galactosidase again is going to go for protein that helps the cells to get identified with through a process called as blue-white screening. Right, now what happens is very simple. I have made it very simple. So what happens, obviously, you can consider this as gene A, you can consider this as gene B. Due to the presence of gene B, gene A gets inactivated. So the gene A here is ampicillin resistance, and this is nothing but beta galactosidase. Right, so what happens, the one resistance which the plasmid was providing to the cell, ampicillin resistance, will not happen now. Right, so what will be the option? Will be able to produce novel protein with dual ability. Right, so this doesn't fit the concept. Second, will not be able to confer ampicillin resistance to host cell. So I think this is sensible. Will not be able to confer ampicillin resistance to host cell, right? Due to the presence of galactosidase. And transformed cells will have ability to resist ampicillin as well as produce beta. So it cannot happen. So beta galactosidase will be functional, but the ampicillin resistance cannot happen, and will lead to lysis of host cells, is also not a good option. So we can go for the option B. Right, so again you could see this, what insertional inactivation. Okay, so the concept has been given, and I said, as in case of previous 1 and 2 question, this is not a direct question. So from this paragraph you need to understand what insertion inactivation is, then you have to apply, and it is better that you know the structure of PBR322, right? Then you have to apply, which makes you to go with the option B. So quite a simple question, application, but quite a simple question, right? Fine. So again, you can see the page number 200 in chapter 11. Okay, 12. I mean, biotechnology 12 series, right? And with respect to this, you can also expect a question, in coming years from PBR 322 chapters. Okay, and I think I forgot a point. See here it has got a PST1. So this is a PST1 is nothing but a restriction site for restriction enzyme. I believe you all know what are restriction enzymes. So restriction enzymes are nothing but an endonuclease, which breaks the internal phosphodiester bond at a particular sequence. So an endonuclease whose activity is restricted is a restriction endonuclease. Right, so the ampicillin resistance gene is going to have a site for PST1 restriction enzyme. So when you use that particular enzyme, a break will be created in which the beta galactosidase will be inserted. Okay, so that is also another concept involved here. So without restriction enzyme, there is any restriction enzyme site, this insertion inactivation is not possible. So before you insert, you have to create a break. So how do you create a break? You have to use a restriction enzyme, right? I hope I am done with this question. And you have to understand what insertion inactivation is, and you have to apply the concept in the above question, right? So fine. And this insertion inactivation, blue-white screening, people might play with these concepts. So what all we need to know is we have to understand it in the basic concept. Once you have understand it, whatever form the question comes, we'll be able to apply those concepts and then get the correct option, right? Fourth question. Nowadays, it is possible to detect the mutated gene causing cancer by radioactive probe to hybridize its complementary DNA in a clone of cell, followed by its detection using autoradiography. Because so this question is again a direct question. So if I'm going to show you this page, you will understand that this is a direct question, again from chapter 12, that is biotechnology application. So I would like to throw some basic idea before we move on to this reference part, so that you will be able to understand it better, I believe. So what is that? Actually, this is something related to a concept called as southern blot. Okay, so I just try to summarize what southern blot is. So southern blot, you say, for example, if I give you a set of DNA, maybe 10 DNA. So DNA here I mean something which is made up of nucleotide. So don't exactly imagine the double-strand helical structure. So in biotechnology, the very word DNA means something which is made up of nucleotide. So I give you that, right? So what do? You have to blot it. You need to find whether you are a particular gene of interest. You are interested in a particular DNA or gene. So I keep using the word DNA gene. So here both means one and the same. Whether it is present or not, you need to know your DNA. So what do? You go for, maybe you can go for a southern blot, right? So how do? You try to do separation. Okay, you go for a separation, and you can go for a blotting, and you can go for detection. So these three are the wings of southern blot: separation, blotting, and detection. So separation is usually done by agarose gel electrophoresis. Then you transfer the separated DNA into a nitrocellulose membrane, right? Then with which you try to do the detection process. So here to detect, we are going to construct something called as probe, right? Probe here means you have a complementary nucleotide, COMPLEMENTARY. So set of complementary nucleotide with radioactive labeled isotope. Some radioactive material attached to the complementary nucleotide. Complementary to what? Means complementary to the DNA which you are looking for, right? So for example, if my DNA which I am looking here is, say for example, AGCC, then my probe will be TCGG, which is attached with the radioactive material. So if I'm going to add this probe TCGG, it will try to look for this particular sequence, which will try to bind. So once it bound, then the radioactivity, which is there in the probe will be emitted, and I'll be able to detect the radioactivity through a technique called as auto radiography, right? This is what, okay, usually is done in southern blot, right? So now what do they say? The question. So this is the sequence. In case if there is a, say for example, there's a change in sequence, say mutation. So mutation is nothing but the change of nucleotide sequence. Say AGCC becomes ACGC. The probe which I'm adding cannot bind with this particular sequence. So there is a mutation happening here, right? The probe which can bind with the sequence before mutation cannot do it after the mutation. Why? Because the complementarity or the pairing is lost, right? So that's the basic. So if this probe doesn't bind to that particular sequence, then the radioactivity will not be seen. So you cannot detect the sequence through autoradiography. So your autoradiographic film for that particular DNA will be blank, right? So I'm trying to make it, this is basically to address the questions, related concepts. So I'm trying to make it as simple as possible. In case if you have any doubt with these concepts, do not hesitate to comment or ask me. I'll be happy to, make further clarification on these things, right? So with these things, so nowadays it is possible to be mutated gene. So you detect a mutated gene causing cancer by allowing radioactive probes. So you know the sequence of the mutated gene. For example, let me assume this is this is the gene just got mutated to this, and now you know the original sequence. So what mutation happens, we are quite unaware of. It can be anything, but we know the original sequence. Okay, by allowing radioactive probe to hybridize to its complementary DNA in a clone of cell, followed by its detection using autoradiography. Because, okay, so actually once it got mutated, your probe doesn't bind to the your DNA that you are looking for. So here it is the DNA for cancer, right? Gene for cancer. So mutated gene doesn't appear on photographic film as the probe has complementary with it. So if a probe has got a complementary, then you could see that. So it's given the other. So one part of the statement is right, option is right, but the other part seems to be wrong. So you cannot go for this option A. Mutated gene is partially appears in photographic. Nothing like partial or full. So this is also totally wrong. And C, mutated gene are completely and clearly appears on photographic film. No, cannot happen. So you are, since due to mutations, I make the message clear once more. Since due to mutation, your probe doesn't bind. Okay, so your probe doesn't bind means no radioactivity. Radioactivity will be exhibited, will be released only if the probe binds the complementary sequence, which is not happening here. So if there is no radioactivity through autoradiography, you will not be able to detect the DNA, right? This is the concept. So mutated gene does not appear on photographic film as probe has no complementarity with it. So I think this option is sensible. So we can go for this B option, right? So I have given you the basics, okay, to an extent which is not possible. So in case, if I repeat again, in case you have any clarifications or doubt, okay, please do approach me or any of your teachers, okay, whomever you are comfortable with. And you might, maybe there might be from the same concept, there might be another question in coming years. So it's better that we need to understand the concept, not just the lines, okay, but the concept associated with each and every line should be clearly understood. So if that is going to happen, whatever be the question, whichever form it is given, it will be easy for us to attend with full confidence, right? So coming to here, we have seen the question, we have seen the option, and we have seen the the discussion. So here you can see where it is. See, the clone having mutated gene hence will not appear on the photographic film because the probe will not have complementary DNA with the mutated gene. So this is the line which is the highlighted in the above question. So we know the line. This is again, this is a direct question, like the first and second question, direct line from the NCERT, and as well as the concept of this line, okay, we have discussed, okay, in a very brief manner, right? So I think we have done with this question 4th and next is 5th. So this is based on, again, molecular diagnosis, in particular Southern blot. So chapter 12. Okay, fine. So next is, this is another question from PCR. During the process of gene amplification, so the what is that? The answer for one of the previous question is given in here. The question, in this question. So during the process of gene amplification using PCR, if a very high temperature is not maintained in the beginning, then which of the following steps of PCR will be affected first, right? So you have to go back to this diagram again. So we have already seen PCR has got 3 steps. There is a step of denaturation, annealing, and extension. There are 3 steps. So first is denaturation. So denaturation, generally you supply a heat with a temperature of 95 degrees Celsius. Huge amount of heat. Why do you do that? So the 2 strands of DNA are in turn attached by a bond called as hydrogen bond. There is a hydrogen bond sitting between the nucleotides of 2 strands. So when you supply heat, this heat will break the hydrogen bond. Say if this is going to be 5 prime 3 prime, this is going to be 3 prime 5 prime. So this heat heat induced disruption of hydrogen bond will result in separation of these 2 strands. So once this happens, then your primer will be able to exactly bind with the specific gene. Okay, so here is what is heat applied in PCR, and that's the reason why do we go for Taq polymerase. We don't use the normal polymerase in PCR because normal polymerase is not heat resistant, because we need to have an enzyme which is supposed to tolerate or which is supposed to withstand the high temperature. So thereby we use a special polymerase from a thermophilic bacterium called as Thermus aquaticus, which is what is called as Taq polymerase, right? So which means that the question here is, during the process of gene amplification using PCR, if a very high temperature is not maintained, we don't maintain a high temperature in the beginning, it's very clear in the beginning, then which of the following steps of PCR will be affected first? So based on the discussion, we can very well go for the option denaturation, right? Denaturation is the step in which temperature induced breakdown of hydrogen bond between the opposite strand of DNA will happen. So here DNA means double stranded helical structure, and you can very well omit the option ligation. So ligation is not at all a part of PCR, right? So whereas the annealing, extension, okay, they are the steps in PCR, but ligation is something different. So quite simple. So you already seen there are almost 3 questions, which have come from polymerase chain reaction, right? Next is question C. So this question is basically from biotechnology application, how to make a genetically engineered insulin. So genetically engineered GE insulin means genetically engineered insulin. So genetically engineered insulin means recombinant insulin. Why do we use a recombinant insulin? What is recombinant insulin? Recombinant insulin is insulin. Sorry. Recombinant insulin is an insulin, human insulin, okay, produced through rDNA technology, that is using biotechnology. We try to get these hormone in a bacteria, to make it very simple, because almost due to various reasons, most of our members of the society suffer from diabetes mellitus. So many of them are totally dependent on this sort of artificial insulin to maintain their blood glucose level. So this is very important, right? Very important in of biotechnology in pharmaceutical industry, application of biotechnology in pharmaceutical industry. So this is all, the question is all related to it. The question is, with regard to insulin, choose the correct option. C peptide is not present in matured insulin. So this is again a straightforward question. You can see that this is a matured insulin. Okay, they have given, actually the question is, they have given 4 options. They have to choose the option which has the correct statement. So that's the idea here. So you can see that the C peptide is not present in matured insulin. So this is the matured insulin. Where is it? This is the matured insulin. From pro-insulin, you have got a matured insulin which has got no C peptide. So the C peptide which is there in the pro-insulin is removed. This is the C peptide, right? It is removed. So I think this statement is true, right? The statement is true. B statement, the insulin produced by rDNA technology has got C peptide. So it is quite contrary to the first statement, right? So this statement is false. Here what do they mean? Insulin is matured insulin, right? And the pro-insulin has got C peptide. Yes, the pro-insulin has got C peptide. So this statement is also true. So you can refer the diagram. I think I should have mentioned, yes, this is chapter 12, page number 211. You have got this diagram and relevant information. How do you make genetically engineered insulin? Okay. And the D option is a peptide and B peptide insulins are interconnected by disulfide bridges. So you can see this. So this is nothing but the disulfide bridges. I'll make it clear. So disulfide bridges. So A peptide, B peptide. There are 2 subunits in insulin hormone. You'll be aware of A and B with 20 and 31 amino acids. So total number of amino acids in an insulin protein will be 51, if I'm right. So the 2 chains are connected by disulfide linkage. So this statement is also true. Okay, so the one statement which is not correct here is B. So you can go for ACD only. So this again, and another direct question from NCERT text. So this question is done. Fine. So next is 7th. A specific recognition sequence identified by endonuclease to make a cut at specific position in the DNA is. So this question is based on the concept restriction endonuclease. Restriction endonuclease, right? I have already described what endonuclease is, one or two questions before. That is, when we are discussing about the plasmid question, the insertion inactivation based questions. I have, while discussing the PST one, would have given what an restriction endonuclease is. Again, to repeat it here, they are basically endonuclease, which means that they cut the phosphodiester bond. What is a phosphodiester bond? It is the bond that connects between two adjacent nucleotides. The one H bond which you have seen before, few minutes before, is the one which connects the opposite nucleotide. Okay, you have got two strands. So opposite nucleotides are connected by H bond, adjacent nucleotides are connected by phosphodiester bond, right? So restriction endonuclease. So this mainly targets phosphodiester bond, and the word endo means it targets internal phosphodiester bond. So that's the meaning of the word endonuclease. And if you consider a DNA, it has got many nucleotide, internal nucleotide, right? N1, N2, N3. If I'm going to start it with N1 to NN, it has got many nucleotide. So of which, if this restriction endonuclease is not free to attack or break any internal phosphodiester bond, which means that we are going to restrict its activity, then in the endonuclease will be referred as restriction endonuclease. You're getting the point. Restriction endonuclease has got properties to be discussed. It has got a nomenclature. It has got the one or two types of products, restriction digested products, of which one important property of restriction endonuclease is it has got a recognition sequence. It has got a recognition sequence which are basically palindrome. Which are basically palindrome. Say, for example, the very common example that we normally use here, even the one which is given in our NCERT text, is the word Malayalam. 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 It is the name of a language of Keralites, Malayalam. So this example is used to understand what palindromes are. What is that? If you read from this direction to this direction, you will get the same sequence of words, and you're going to repeat it in the reverse, you get the same sequence of words. So Malayalam or madam, you can also take MADAM, madam, get the same. Okay, you can try. So this is nothing but a palindrome. So endonuclease recognition sequence are basically palindrome. So we have a particular sequence in which the endonuclease is going to cut the internal phosphodiester bond. So to demonstrate it, let us try. If we take the very common restriction endonuclease EcoR1, 5 prime GAA TTC 3 prime and 3 prime CTT AAG 5 prime. So I hope I have given the correct sequence. Sometimes I think it's a correct sequence. EcoR1. So if you see, if you do it from this direction, you get the same sequence. If you are going to do it in the reverse direction, you get the same sequence. So they are basically palindromic, which is one important property of restriction endonuclease. So poly A tail sequence will not be the correct one here. Degenerated primate sequence, Okazaki sequence. So you can go for the palindromic nucleotide sequence. So the one option which can be the correct one, right? So this is 7th. So and you can see this palindromic nucleotide sequence. The reference have been given in page number 196, chapter 11. So this restriction endonuclease will be the part of biotechnology principle, right? Fine. And 8th question. During purification process of rDNA technology, the addition of chilled ethanol precipitates out. Fine. Again, this is from principal chapter. In principal chapter, we do have a topic called as DNA isolation from a cell. So even I have given a video on it. If you want, you can please go through it so that you'll be able to understand this DNA isolation more clarity. So I'll try to summarize as far as possible. You have a cell. That is a cell from which I'm going to isolate DNA. So what can be the cell? It can be any cell with one condition. The cell should be nucleated. Right? Cell should be nucleated. So enucleated cells, the cells from which the nucleus is removed or nucleus is not there, you cannot use it here because without nucleus, they don't have DNA. So bacteria also have a nucleoid in primitive nucleus. So the nucleus with DNA or nucleus-like structure with DNA should be there for you to do this. RBC platelets in case of humans cannot be used because they don't have, matured RBC, they don't have nucleus, so no DNA. So I have two steps here from which, to isolate DNA from this, I have two steps. One is I need to target the membrane. Quite simple. I need to target the membrane. The membrane will vary from cell to cell. So in case of plant cell, there's going to be a plasma membrane with cell wall. Same with fungi. Same with bacteria. But in animal cells, there are no cell wall. So you have only the cell membrane as the outermost membrane. Right? So to target the membrane, we need to add cell lysis. So lysis is something break, which is going to break the cell. So cell lysis solution. Cell lysis solution. The component of cell lysis solution will vary from cell to cell. So in case if I need to target, a plant cell to break the plant cell membrane, I have to add cellulase because the content in the plant cell wall is cellulose. In case of fungi, I have to add chitinase. Hope you get the point. So once I do that, if my wall is all broken, my nucleus will be, nuclear content in turn will get disintegrate. So all the cellular components will be released into the tube in which I'm doing the experiment. Once I break the cell. So once you break the egg, you get all the contents into the vessel. Likewise, here you get all the contents of cell into the tube. So this is the first step, of which among all the contents, I have my DNA because my idea is to get the DNA out of the cell. So to concentrate, to precipitate my DNA and leaving all the contents back in the tube, I'm going to do the second step, which is DNA concentration, or we can also call it as DNA precipitation. So how do you do it? Just add chilled ethanol. Okay, chilled ethanol. So this addition of this chilled ethanol will try to precipitate the DNA, leaving all the contents behind. You can just take the DNA and use it for your further purpose. So this question was asked actually, I think 2018 NEET has got this question, but in another way. They have given, in order to isolate or precipitate DNA, which is the chemical that can be used. An option was chilled ethanol. One of the option was chilled ethanol, which is the correct. And now they had made that option as a question, and they have given the question as an option. So just ulta, but concept is all same. So this is all written. So you can very well go for DNA, right? And the necessary information is given in this. Again, this is a direct question from NCERT text. You have got it in chapter 11. So I think the lysozyme, cellulase, chitinase, everything is given. Cell lysis solution and chilled ethanol is given here, and you could see this is the DNA. Right, so page number 201 has got the relevant information. So kindly go back to that and get the concept. Right, fine. So next is 9th one. DNA strands on a gel stained with ethidium bromide when viewed under UV radiation appear as. Right, fine. So this is from the topic, concept agarose gel electrophoresis. So again, this question was asked previously, the same concept but in a different form. What stain do you use to visualize the DNA in an agarose electrophoresis? And one of the option was ethidium bromide. I always say to my students, there is nothing new that is invented in recent days. So if you are thorough with some basic concept that is given in book, or if are the related questions, you would find your paper easy. And that's what has happened. So many people were able to cross 300 this time in biology. There is the reason. It's true with the other subjects also, but what you need is more practice, with concepts and related problems, right? So agarose electrophoresis. So here, what do we do? As we have seen previously in the southern blot, I think 3 to 4 questions before, we have seen, you have an agarose gel in which you are going to separate your DNA. Say, for example, you have some 6 to 7 DNA with you or gene with you, you want to separate it. You do it by agarose electrophoresis. Electrophoresis is a technique where a charged molecule can be moved, okay, can be allowed to move, which is the movement is based on principle E by M. So the movement is directly proportional to charge and inversely proportional to mass. So more the mass, less the movement, less the mass, more the movement. So you have got, say for example, you have been given a tube with 8 genes or DNA. You are asked to separate it. You allow it to do by agarose gel electrophoresis. So once the separation is done through agarose gel, the question is, why do you use an agarose gel? Means that the any movement for it to happen efficiently, the supporting medium should be inert, which means it should not interact with the substance that moves. And agarose is going to be inert to the DNA. There is no interaction between the moving DNA and the supporting agarose gel. So hence we use agarose gel. So in case in future, if you're going to get any other material which is going to do the same role, probably if it's going to be cost effective, we can also use it. Who knows? So if the science will provide a different thing, effective thing. But agarose is quite effective. There's no doubt about it. It's very comparatively cheaper and more effective, right? So we do it. So once we do it, the question is whether the DNA movement is visible. Okay, which means that no, DNA is not a chromogenic. Chromogenic means it won't produce any color. DNA is there, but it is not visible because we have separated the DNA. So how we have to somehow make it visible. So how do we do that? You add something called as ethidium bromide, a stain called as given ETBR. So I make it in the short form ETBR. You add ethidium bromide. Ethidium bromide is basically a fluorescent dye, which means that you try to smear the ethidium bromide throughout the gel. So this region doesn't have any DNA. This region doesn't have any DNA, but this region has got DNA. So wherever the ethidium bromide is going to bind with the DNA, it is going to form ethidium bromide DNA complex. So once the ethidium bromide complex itself with the DNA, this is going to get fluorescence. You're getting the point. So DNA on its own is not fluorescent, which means that there is no need for this step. Ethidium bromide on its own is not fluorescent on its own, but when DNA combines with ethidium bromide, the DNA is going to increase or augment the fluorescence of ethidium bromide. So what is fluorescence? You have to understand that. What is fluorescence? Fluorescence is something you provide an incident light. A fluorescent dye will try to use that incident light, and you get a emitted light in a higher wavelength. That's what is fluorescence. So for which you have to provide a light in UV region. Okay, ultraviolet light is what is required here. Why is ultraviolet required? Is all a different part of discussion. So you give an ultraviolet light, and you give a sort of orange, right? Orange light, orange bands. Okay, so which means that there's a bright orange band is the option here, right? I can show you this diagram. So this is what is the bright orange. So this is the entire gel what I have shown. Here also there is an ethidium bromide, right? But here is the ethidium bromide DNA complex. So this is going to increase the fluorescence of ethidium bromide. So you give an ultraviolet ray, in return you get a band in a orange region, and you have got an I think it's a direct question. So exposed to UV light, orange colored bands of DNA in an ethidium bromide angel. So you can see the option cited here. So bright orange band. This is 9th question. Another straightforward question from agarose gel electrophoresis, and agarose gel electrophoresis will be from biotechnology principle. Right. So the last question, I think, the 10th question. When a gene targeting involved gene amplification is attempted with in an individual tissue to treat a disease, it is known as. See, this is something from the concept gene therapy. So what is gene therapy? There is a genetic disease. So what is genetic disease? A disease that results out of an improper gene function. The gene is at error, maybe due to mutation or something, the required function of gene is not being carried out due to some reasons. So rather than, if for example, the best example that you can consider, diabetes mellitus. Diabetes mellitus type 1, where the insulin gene in the pancreatic beta cell is somehow not functioning. There is a mutation or whatever be it. So in insulin protein, which will be coded by that gene, is not produced. So what do we do? As we have seen earlier, we give the patients with artificially, there is recombinant insulin hormone which is produced through biotechnology. The very word recombinant means it is produced through biotechnology, will be administered as a treatment option. So rather than giving it as a protein, here in gene therapy, what do? We try to directly target the gene. Beta cells, the defective insulin gene will be replaced with the normal gene. Okay, so that is what gene therapy targets. So any disease where the gene is not functional due to any reason, we try to replace the error gene with a functional gene, or non-functional gene with a functional gene. So this is what is called as gene amplification. So here gene amplification doesn't mean the one which is, we usually do it using PCR. So here we try to increase the number of gene, replacing the error gene. So which is what is called as gene amplification, or you can also call it as gene augmentation. The other one is in case if a gene which need not get expressed, whose product is not required. Say for example, a cancerous cells will have a gene called as oncogene. Let me consider this as an oncogene. This is going to code for an oncoprotein. Okay, this oncoprotein is going to make the cell cancerous. Okay, so I don't need this oncogene to get expressed. So we can also block the oncogene through RNA interference or RNA silencing. This is also sort of gene therapy. So we try to either activate a gene, or we try to inactivate a gene based on the situation. Okay, directly the gene. The process here will be referred as a gene therapy. Therapy means treatment. So treatment by directly activating or inactivating a gene. Okay, so this is all regarding gene therapy. I think the option is quite clear. So gene therapy. Okay, so molecular diagnosis is not the option. Biopiracy is something different. Safety testing is different. So gene therapy. To treat a disease, so therapy. Okay, gene targeting. Right, fine. And you have this information here at chapter 211. I mean, chapter 12, page number 211. The first paragraph gives you the basic details of what gene therapy is, and this information is very important. The first gene therapy, when was it done, to whom it was done? Okay, so there's something called as SCID, severe combined immunodeficiency disease, which is due to the deficiency of enzyme adenosine deaminase. Okay, so this was the first case which was treated using gene therapy. So gene therapy might be a good weapon against treating various genetic diseases in near future. There are some issues that needs to be resolved. Once it is done, gene therapy will be a very good option for treating genetic diseases, right? So I think we have done with the questions. So there were 10 questions from Unit 9, Biotechnology, 10 questions. So I think this table will give you the concepts from which the questions have come. So you got a topic, chapter, and number of questions. So PCR has got 3 questions. You can note that PCR has got 3 questions from chapter 11. And the molecular diagnosis has got 1 question. Selection process, there is insertion and activation, has got a question. Genetically engineered insulin has got a question. Restriction endonuclease has got a question. DNA isolation has got a question. There is chilled ethanol, agarose electrophoresis, gene therapy. So almost all topics of the important topics of these 2 chapters are covered in the 2021 paper. So what all we need is, we need to be thorough with each and every line and each and every concept, okay, given in the NCERT text. Generally, people say you have to be thorough with line to line. So line to line thoroughness is important, but it should not be just remembering those lines, but you should also remember the concepts associated with each and every line. Okay, I mean, which are each and every ideas, right? So totally we have got 10 questions. So when we understand the concept with those lines, then obviously our attending the answers will be much more simpler and easier, more interesting. More than simple, it will be more interesting. So it will definitely take your marks towards 350, 360, or more than 340. 360 will be the maximum mark that we can get. So it will take your score towards 360. Right. And this is the final thing that I'd like to show. It has got 6 questions from biotechnology principle. There is unit, as I said, unit 9 has got 2 chapters and 4 from biotechnology application. So biotechnology seems to be a sort of difficult chapter compared to the other chapter. The difficulty here means there should be a proper, as I said, there should be a proper understanding in the various concepts given in the biotechnology principle. If that is being done, then these questions will be very easier to crack, right? So totally there are 10 questions. So I hope this video would have been useful to you. So I'd like to continue this with the questions from other chapters too, like human physiology, plant physiology. Maybe you might be getting in very near future the question discussion from other chapters. So I'd like to have your feedback in the comment section, or you can even mail me. So your valuable feedback, healthy criticisms, everything will be required, to improve the quality further. Any suggestions are also welcome. So please do subscribe to the channel, and please share this with your friends. And in case if there are any doubts, okay, in the concept that we have seen in this question paper analysis, please do feel free to ask me. I'll be happy to help you. So with all these words, thank you so much. We'll meet soon in another video. Thank you.